When we look at the cross section of the spinal cord, we again notice that there is a H or butterfly shape in the middle, and it's surrounded by white matter. And that is not by accident. There is a very specific organization of gray and white matter. And if you remember from chapter 14, chapter 15, we discussed why it has this color. Why it has this color, again, is because in the white matter, we have myelinated axons, whereas in the gray matter, it's mostly cell bodies, dendrites, unmyelinated axons. While we don't discuss the specific path of various pathways from the brain to the spinal cord and out to the effectors, we can at least understand that in this shape, in this shape right here, that we have, it's a different color because we don't have lipids, whereas in the white matter, there's a high lipid content due to the myelination. Now, when we look at the cross section, there are regions of white matter and gray matter, and we'll see why. But the naming of the white matter are called funiculi. One is called funiculus or a funiculi. They're also called columns. Yeah, so this posterior region, you have both a left and a right side. On either side of the posterior median sulcus or dorsal median sulcus, you have a right and a left posterior funiculus. You have a lateral funiculus, or it could be called a lateral column. You have an anterior funiculus or an anterior column. And then in the middle, crossing over from right to left or left to right, is this white commissure. And we're going to attach some vocabulary terms, or I think we did in chapter 15. We talked about various tracts, and we talked about what a commissure is. And a commissure, by definition, is a tract that serves to connect the right and left side of the brain. We also have it in the spinal cord. When we look at the gray matter also has regions called horns. So you have a posterior horn back here, an anterior horn or ventral horn here, and then the lateral horn, which typically appears in the thoracolumbar region. Hint, hint, wink, wink. Wonder why there's a lateral horn only in the thoracolumbar region. And then if we look at an image, histology image, we can also still see this very clear butterfly shape. We can see the posterior horn, lateral horn, and anterior horn, as well as both the gray and white commissure. And then right in the center of the gray commissure is this central canal where we have flow of cerebrospinal fluid. Sensory and motor information is segregated in the spinal cord, and the cell bodies of both sensory and motor are located in different regions. Now we identified this structure right here, which is the posterior root ganglion or dorsal root ganglion. The dorsal root ganglion or posterior root ganglion contains the cell bodies of sensory neurons. So sensory information, afferent information, enters the spinal cord via the posterior root. Motor information is going to exit, efferent information is going to exit the spinal cord via the anterior root. When we talked about functional subdivisions of the nervous system, we talked about two types of sensory information, and we talked about two types of motor information. We have somatic sensory information is information we are aware of. Visceral sensory information is information we are not aware of, just as a review. And then we have two types of motor: autonomic motor, which we cannot control, and then somatic motor, which we can control. And we know that the one target of somatic motor neurons are skeletal muscle. So that information is very well organized when we look at the cross section of the spinal cord. In the posterior region, the posterior gray matter, the posterior horn, we have sensory information. We have sensory cell bodies projecting into the gray matter, and then we have interneurons, sensory axons and interneurons, and they're separated by somatic and visceral sensory. And then when we look at the, did I just say posterior? Posterior. When we look at the anterior side, we have motor axons and motor axons and motor neurons. And they are also segregated by autonomic and somatic. So you have autonomic motor neurons that are more found on the lateral horn, and then you have somatic motor neurons that are found here in the anterior. Another thing that we notice is that there is a predecessor. There is a wire, which we saw much earlier in the semester. We saw roots and rootlets coming out of the spinal cord. So we have, I should actually change the direction of that arrow. We have sensory roots entering the spinal cord this way, and we have motor roots exiting the spinal cord that way. And then they eventually all merge to form what's called a spinal nerve. When we looked at what a nerve is made up of in chapter 14, we saw that there's connective tissue covering, that we have axons with myelination, and we have this connective tissue covering that brings together many thousands of axons to form a single spinal nerve. So what we see here is that the spinal nerve is actually formed in the intervertebral foramen, which we saw in the thing right here. So when we look at our spinal nerves, we have three connective tissue wrappings, just like we learned in chapter 14: endoneurium, perineurium, and epineurium, and that is from deep to superficial. And we use the same kind of naming mechanism that we used for our connective tissue covering in muscle. So again, we have this spinal nerve here. This is the spinal nerve here. Now, what I want you to realize about the spinal nerve is that the spinal nerve contains many axons of both motor and sensory neurons. So the roots, or the root, we have little tiny rootlets that merge into roots that emerge, get out of here, that emerge from the spinal cord, either anteriorly or posteriorly. They come together in the intervertebral foramen. And we have the cell bodies of the sensory neurons right here posteriorly, but then they come together to form a spinal nerve. The spinal nerve then branches into three wings, or three rami. You have a posterior ramus, an anterior ramus, and a rami communicans. And they look very different. They are colored, not colored, they are sized very differently in this stylized cartoon. What you see is the posterior ramus. What is it going to do with its life? It's going to innervate the deep muscles of the back, like the erector spinae muscles. So it's a very small branch that contains both sensory and motor axons and goes and innervates deep muscles of the back. Then you have this anterior ramus. It's thick, thick. She is much larger than the posterior or the rami communicans. And the spinal nerve, this whole spinal nerve, this one wing of it, the anterior ramus, is going to go and form a braiding called a plexus, a spinal plexus. It's going to go braid itself up, and then named nerves that go and innervate regions of the skin and muscle are going to exit from those plexuses, and we'll take a look at them. And then lastly, we have this rami communicans, this little tiny guy, and the rami communicans contains, you guessed it, sympathetic. It contains axons of sympathetic neurons, autonomic neurons that emerge from the thoracic and lumbar region of the spinal cord, and that are located, their cell bodies are located in the lateral horn of the gray matter of the spinal cord. Okay, so again, we have rootlets emerging from the spinal cord. It's different information. If it's posterior, then it is sensory information or dorsal, and then if it's anterior or ventral, it's motor information. We know that information is directional. Sensory information is coming in, motor information is going out. And those roots form a spinal nerve right at the level of the intervertebral foramen. And then each spinal nerve, we said there are 31 pairs of spinal nerve, one on each side, right and left side, are going to split into three branches called rami. We have a posterior ramus, an anterior ramus, and a rami communicans. Yay. Words. Dermatomes are really cool patterns of innervation from spinal nerves to the skin. And they just so happen to innervate in a pattern based on where that spinal nerve comes from, except for C1. So a dermatome is a map that follows a segmental pattern around the body. And they are clinically significant because if you have anesthesia in a particular region of the skin, it can indicate spinal nerve damage, right? It's quite an investment to cut a human body open and do neurosurgery. So, and it's also not very accessible, right? We have the brain in the bony cranium. We have all of those meninges. We have that blood-brain barrier. We have CSF. Same with the spinal cord. It is enclosed in a bony cage, the vertebral column, or the spine. And then you have all of these meninges. So it's quite handy that there is a pattern of innervation on skin that tells us something about spinal nerve function. We also have referred visceral pain. So if you have a dermatome that is innervated by the same spinal nerve as, let's say, the appendix or the liver or something else, that can tell you something about that liver, that spinal nerve function. So here is a map. It's an approximate map of where each spinal nerve innervates on the skin. And what we'll see is that spinal nerves, again, are mixed nerves. They have sensory and motor axons, and they do both sensory and motor information. So if you have, for example, and skin is something that you can feel, right? This is sensory information. T10 around the umbilical region, if you have numbness around T10, that could tell you something about the T10 spinal nerve. Now, once that spinal nerve exits the intervertebral foramina and it forms that braid, it forms those three wings, your anterior ramus, your posterior ramus, and your rami communicans. It's the anterior rami that come together and form a plexus. And a plexus is literally, the term plexus means a braid. And the nerve plexuses are what give rise to the spinal nerves that we name, that go and innervate all of those skeletal muscles that you named and you memorized their actions and their attachments in lab. Okay, so we're going to look at a few spinal nerves and we're going to look at the plexuses that they emerge from. We have four major plexuses. We have a cervical plexus, a brachial plexus, a lumbar plexus, and a sacral plexus, and we will practice remembering them in lab. We don't have a thoracic plexus. Why do we not have a thoracic plexus? Well, if you think about where along the spinal column that these thoracic spinal nerves emerge from, it's from the thoracic region. The thoracic region is where you have your thoracic cavity, ribs, breathing muscles, intercostal muscles. So the intercostal nerves that emerge from the thoracic region of the spinal cord don't form plexuses. Instead, that anterior ramus, you still have the posterior ramus going and innervating deep muscles of the back. And you still have that sympathetic trunk. You still have these ganglia. I don't think I even mentioned these ganglia, right? We mentioned rami communicans contains axons of autonomic neurons, but they connect and their cell bodies are located in sympathetic trunk ganglia. And we said ganglia are part of the peripheral nervous system because they are outside of the brain and spinal cord. So back to my original point here is that the intercostal nerves that emerge from the thoracic region of the spinal cord go directly and innervate intercostal muscles, the breathing muscles, the internal and external intercostals. Okay, is that the best I got? That's the best I got. So if we look at the what a plexus actually looks like, and I actually do not want you to know the branches. You do not have to memorize the branches, but they are. It looks in the stylized cartoon. It looks like this. Here's your spinal nerve right here. Oh Lord, here's your spinal nerve in orange, right? It is a merging of motor and sensory roots that come together and they form a spinal nerve. And then the spinal nerve is going to split and it becomes, it splits into the rami. Here's your anterior ramus. And it's got all of these different branches. And then here in yellow is where your spinal nerves come out of. When we think about why the heck something like this happens, it's quite interesting, right? Why not just have a direct, just like one cable coming out of the spinal region and going and innervating a particular region in the gut, let's say, or in the upper limb, let's say. Well, it just so happens that if you have damage to a spinal nerve, you don't completely lose innervation of its target because of this braiding, because of this redundancy of axons. We have thousands of axons coming together and they merge into a particular nerve and then they split into a different nerve. So you have this redundant way of innervating muscles, muscles of the limbs, as well as internal organs. So when we look at the 1, 2, 3, 4 plexuses, the cervical plexus from the neck is going to emerge from, or it is formed by, I shouldn't say emerge from, it is formed by the spinal nerves, or the anterior rami, to be more specific, the anterior rami of spinal nerves C1 through C4. Then you have the brachial plexus. This is the brachial plexus right here, and it's quite large. It's huge. And the brachial plexus is what innervates the upper limb. That is going to be formed by the anterior rami from spinal nerve C5 to T1. And then you have the lumbar plexus, which innervates the anterior and medial thigh. We'll do a whole little video on those. And that emerges from, or that is formed by, the anterior rami of L1 through L4. And then you have the sacral plexus, which gets the rest of the lower limb, and we'll go over it in a drawing video. And that is formed by the spinal nerves from L4 to S4. Yes, you do need to know these, because we're going to see what regions of the upper limbs and lower limbs are innervated by each of these plexuses.